Hi, at Lab 4 Enzymes, we're going to be looking at how enzymes help break apart substances in our digestion process. The objective in this activity, you will conduct an investigation that teaches the effectiveness of an enzymes under different conditions. Introduction. Enzymes are catalyst chemicals reactions. In digestion system, enzymes help break down proteins, carbohydrates, and fats into smaller molecules. Carbohydrates are broken down into simple sugars such as glucose. In this activity, you'll test the effects of lactase enzymes on two types of sugar: milk sugar, lactose, and table sugar, sucrose. Also, you'll test the enzyme's effectiveness in the different environments. So right here is just the scoring of how the breakdown of how you'll be scored. Enzymes. In this activity, you'll follow Dimitri, a fellow biology student, as he tests the effects of lactase enzymes in different conditions on different solutions. This should take about 2 hours to complete. It could take a little bit less, could take a little bit more depending on it. So if you were doing the experiment, it gives you a breakdown of the materials you'll be using, and it will break down basically what you'll be testing, which is milk and water, so lactose and water, milk, lactose, and lactase in water. And this is the enzyme. Then you have milk, lactase enzyme, and water, but the water has been heated to a boiling point. And then in test tube number 4, you're going to be testing sugar and water. And then test tube number 5, you'll be testing sugar and lactase enzyme in water. Then in test tube number 6, you'll be testing sugar and lactase enzyme in water, but the water has been heated to boiling. So right here, it basically will say each solution will be in a test tube, and then Dimitri will insert glucose test strips into each solution and test the presence of glucose. The test strips will change color if glucose is present in the solution, as indicated by the following scale. Now, the one kind of caveat or disclaimer I want to put in there is this blue color. This bluish color is really more bluish in this picture than the actual test sticks. And so what happens during the filming, where they took the pictures, it's going to show a little bit different color than right here. I'll try to help explain it so you can tell. So what you can do is you're going to create your hypothesis, and you're going to create your hypothesis by looking at these two diagrams. The top part is substances being tested. So you got sucrose, which is just table sugar. You can see how it's broken down. It shows a representation of the two elements being put together, and then lactose on the right-hand side. Now below it shows simple sugar compounds. That's fructose, glucose, and galactose. So you're going to use the simple sugar components and see if it has any of them in the substance you're tested. And then you're going to create a hypothesis using the molecule structure that predicts which 6 solutions will test positive for glucose. Write down your prediction and the answers, provide your reasoning behind it. So you're going to see, and basically test tubes 4, 5, and 6, you have sucrose. And then in test tubes 1, 2, 3, you're going to have lactose. So are sucrose and lactose, are they going to have glucose in them? You're going to use the glucose molecular structure and see if it's inside either one of them and predict, will it be broken down? Will you be able to test for glucose in the solutions? And you can look back up here, solution 1, solution 2, solution 3, solution 4, solution 5, solution 6. And I'm saying solutions that are in the test tube. So you can say test tube 1, test tube 2, test tube 3, test tube 4, test tube 5, test tube 6. So write down your answer or your hypothesis, and then you're going to go to question number 2. Now, you're not going to be able to do the experiment at home, so we have broken down basically the experiment with these pictures. So we're going to be doing the testing in test tube number 1. So basically, the biology student filled it basically 1/4 full of milk and then 3/4 full of water, and that's all that's in it is milk and water. So that's the test tube strip. Now look at the color right here, and you're going to compare that color to this as a result. They put the glucose test strip in there for 30 seconds after they've already swirled and mixed it around with the stirrer. And then they pull it out. If you look at it, even though this color does not perfectly match this, you can see that it relatively matches this, pretty much similar, except that it's wet. So in this case, we're going to say that this is negative. So in test tube number 2, the Dimitri filled it full of milk, about one quarter full, and then 3/4 full of water, plus the lactase enzyme that was a lactase pill that was dissolved in the water. They stir it up, mix it. They put the glucose test strip in there for 30 seconds. Now, if you look this color versus that color, is there a change? Yes, I would say there's a change. And the change goes towards being positive. So I would say in test tube number 1, it definitely is positive. So right here, test tube number 3, the milk with the heated lactase enzyme water milk was added, then the heated lactase enzyme water was put in the test tube about 3/4 of the way full. It was mixed. The glucose test strip was put in there for 30 seconds. You compare these two colors, you can see it looks just like one. So what do you think it is? Is it negative or positive? I'll let you be the judge of that one. You're going to record test tube number 1. Was it positive or negative? Test tube number 2, was it positive or negative? Test tube number 3, was it positive or negative? So when we say it's negative, that means that the test strip indicator means that there's no glucose in there. So that means no glucose was broken down. If it's positive, that means glucose molecule was broken down from the milk, or the milk sugar as it was referred to. So what we're trying to test is the enzyme. Does it work? Does it not work? Does it break down the milk sugar in there into usable molecules that your body could use? And in this case, the only thing that's different is there was heat. Did heat have something to do with the ineffectiveness of the enzyme molecule? That's what we're looking at. So in question number 3, we're going to be looking at this with the sugar. So we have the sugar, just table sugar put in there, mixed with water. Put the glucose test strip in there after it was swirled and mixed for 30 seconds. Now we're comparing these two colors. Was there a color change? And you tell me, is there really a color change? If you don't think there was much of a color change to no color change, then that's going to be negative. Test tube number 5, the sugar with the lactase enzyme water. So we have the sugar mixed with the lactase enzyme water and swirled, mixed around. Put the glucose test strip in there for 30 seconds. Is there a color change from there? You be the judge of that one. If you don't see there's much of a color change, then count it as negative, no glucose present. If you do think it was positive, then you can put positive, but take a close look. If you don't see much of a color change, go with negative. Right here is the test tube 6, which is sugar with the heated lactase enzyme water. So the sugar is put in there. Then the heated lactase enzyme water was also put in there, mixed. The glucose test strip was put in there for 30 seconds. Compare these two colors. Was there a change? If there's not a change, then say it's negative. If you think there was a change, then say that it's positive. So go ahead and record the results. Test tube number 4. Was it positive or negative? Did it have glucose in it? If it's negative, it does not. Then you'll do test tube 5 and test tube 6, record the results. Right here is where you're going to say, did your hypothesis predictions match your results? Yes, no, maybe some of them did, maybe some of them didn't. Go ahead and explain if they didn't or did. And it's okay if your prediction did not match your results. That's why we do these experiments. Using the results from Dimitri's experiment, explain why the color change of some test strips but not others. Now right here is where I'm going to have to break it down for you. That the lactase enzyme is really going to only affect lactose. It's designed specifically to connect with lactose and break it apart. Think about it as a key. You have a house key that you can put in your key lock, turn it, and it will open up. because enzymes are like keys. They go in there and they break apart the molecules that they're designed to break apart. Now, let's say you took your key and you stuck it in a super hot fire that actually melted the key. If you stuck your key after it rehardened into the lock, you probably couldn't turn it. So heat can damage enzymes. So right here, you're going to want to talk about how maybe in the test tube 2 it was positive, but in test tube 3 it was negative, and there has to do with the factor that heat was applied to the enzyme. The other part is you're testing in test tubes 4, 5, and 6 sugar. Sugar is not lactose. So if it's not lactose, it would not actually break apart sugar because it's not lactose, so it can't break it apart. That'd be like taking your key, then stick it into your friend's house lock. It could fit in there possibly. It may not, depending on what type of lock it is. But it definitely would not open up that door, so it would not turn the lock. All the locking mechanisms would not line up. Just like this, the enzyme would see the sugar, but it couldn't do anything to the sugar to break it apart because it's not an enzyme devoted to breaking apart sugar. So right here, we really want to focus on the temperature effect, as I was kind of using the metaphor as if you had your key and you stuck it in a hot fire that actually melted it and damaged it, and you stuck it then back into your key slot. It may not even fit in there. Even if it does, it probably won't line up and work. So heat destroys enzymes. In your body, the reason why you keep at 98.7 degrees Fahrenheit is because if your body gets hotter than that, it's going to destroy enzymes. So heat can destroy enzymes and they won't be able to work. And so in this case, the lactase enzyme is sensitive to heat, which is shown by when you boiled the water and then you tested it and there was no change right there. So that's a negative change on test tube number 3 because the heat destroyed the enzyme. People who are lactose intolerant lack lactase enzymes that often experience digestive problems when the dairy products do the results of Dimitri's experiment support the scenario? Explain your answers. So if you're the unlucky person, then your body doesn't produce the lactase enzyme. You can't break apart lactose, so it's going to be a bigger molecule in your stomach. Now, because it's a bigger molecule, your body can't absorb it, and it can cause irritations. And when your body deals with irritations, it normally causes some type of negative reaction, such as stomach cramping, gas, or constipation. So in this type of situation, and test tube number 3, when the enzyme was damaged, it couldn't break it apart. So it does support the fact that if you don't have the lactase enzyme, you're going to experience problems. How does the results show the digestive enzyme is critical in maintaining homeostasis in your body? Well, you have tons of enzymes in your body to break down all the different molecules that you have, the fats, the proteins, the carbohydrates, as I talked about in the background information. You need all of that, all those enzymes, to break down those molecules so that those molecules can be absorbed into your body through the intestine and go into the bloodstream and then be taken to all the cells of your body so that you can create energy. And that energy used in digestion is what keeps your body running and functioning and maintaining homeostasis. So your heart stays beating, your lungs stay breathing as you inhale and exhale through your diaphragm, and your muscles are able to contract. You're able to see through your eyes. All that is done by breaking down food and then using the broken down food and the molecules to create energy for our body. So hopefully you learned a lot on this, and then you're able to write your answers. Now make sure after you've done everything, you've hit submit, and you've gone to the last one, you hit save and exit, and then your lab will be done. Have a great day, and if you have any questions, give me a call, give me a text, send me an email.